Alright, so now we've moved over to a plant cell. As you can tell, it's got a lot more in common with the animal cell than with the prokaryote. And the reason is because these are both eukaryotes. Plants and animals are eukaryotic, meaning that they both have nuclei. And as you can see, this nucleus, we've got that double membrane bound compartment, just like we saw with the animal. We've got nuclear pores to allow things like RNA to exit. And there's this dark region where ribosomes are assembled called the nucleolus. You can also see associated with the membrane of the nucleus is another tube-like membrane structure, which we call the endoplasmic reticulum. We've got a little bit of smooth endoplasmic reticulum, which just as before is going to be dealing with getting rid of substances that don't belong, storing things like ions that need to be stored away, making substances like lipids. That's what the smooth endoplasmic reticulum is good at. The rough endoplasmic reticulum, what it has are ribosomes that sit right on top of it. The ribosomes are not inside it, they sit on top of it, which give it that rough appearance. Those ribosomes are going to be taking care of translation, just like they were before in both of the cell types we've looked at already, where you're going from RNA to protein. The proteins that might be made in this region on the rough endoplasmic reticulum would be those that belong inside of membranes, so not your cytosolic or cytoplasmic proteins. You want the proteins that are integral membrane proteins. Those kinds of proteins would be made here. You would also make proteins that you wish to export outside the cell, because as you guys know, the rough endoplasmic reticulum is going to package up those proteins that are destined for use in other membrane-bound compartments or destined for life outside the cell. It's going to package them up into vesicles and send them off. Oftentimes, those vesicles end up going to this region we call the Golgi apparatus. And just like you saw before, we're going to have a cis and a trans part of this Golgi. The vesicles will be dropped off at one end, mature a little bit, go to the next part of the stack, mature a little bit, go to the next part of the stack. So this Golgi network is going to be kind of important for getting things like glycosylated proteins together. Once you finish maturing the protein, you're going to package it up into an enzyme again. So this trans region on the Golgi is actually doing quite a bit of business. There's a ton of vesicles budding off of it at any given time. Those vesicles go where they need to go, right? We have some membrane-bound organelles besides the nucleus here, and so maybe they're going there. Maybe those vesicles go to join the cell membrane. That's how you replace old lipids, right? You're going to send them new lipids. And you made those lipids over in the endoplasmic reticulum. And if they needed any sort of modification, that would happen over towards the Golgi. Since I mentioned the membrane, why don't we talk about it? You can see right here that the membrane is pushed right up against the cell wall. That is held in place because we have this large fluid-filled compartment that's primarily water, but also has other stuff happening. We call it the central vacuole. You do some digestion in there, you do a whole ton of storage, and primarily what you're storing is water. The size of that container is very important for pushing everything around the edges, because the membrane is not actually connected to that cell wall at every location. The membrane is only connected at certain specific junctures, which we call plasmodesmata. You can see that here. So these areas, this is where you're going to connect one cell to the other. Because the cell wall is not only going to provide structural support, it's also going to provide a very strong barrier between these two cells. And since you have a pretty strong barrier, how are you going to communicate with your neighbor? You're going to have to literally poke holes. And that is where the membrane would be pretty firmly attached to the cell wall. But if the central vacuole were to shrivel up, then these areas right here would be the only areas still stuck to the cell wall. All of this membrane can fall back in pretty much. So it's kind of important to keep well stocked with water. One of the other structures you see here is a mitochondria. You saw that before as well. It's a double membrane. We've got 3 double membrane compartments in here, actually. We've got the nucleus, we've got the mitochondria, and we've got chloroplasts. Chloroplasts are specialized to plant cells. You do not see them in animal cells. These chloroplasts have a whole lot going on, which we're going to spend a ton of time on when we talk about photosynthesis. So primarily what we care about with the chloroplasts and with the mitochondria is the fact that they have 2 membranes. They also, weirdly, have their own DNA, separate from the DNA for the rest of the cell. So the DNA for a mitochondria, including your mitochondria, is all contained inside that organelle. And in fact, it's circular, which, as you guys know, you don't normally expect for eukaryotic cells. Circular DNA tends to make us think of prokaryotes. The fact that we have circular DNA here is sort of a clue that people have run with and gotten a few other pieces of supporting evidence to come up with this idea, endosymbiotic theory. As you know, endo, this is inside. Symbiotic, we haven't talked about it, but hopefully you remember a little bit about it. Something that's symbiotic, they're living together. So endocytosis would be something like what you can see in the attached link, where an amoeba is eating something, right? That's phagocytosis. It's a version of endocytosis where you take a big chunk out of whatever you're nearby. What we believe happened when we got mitochondria and when plants got mitochondria and when plants got chloroplasts is that there was some ancient form of bacteria with its circular chromosome and double membrane, as some of them have, that got engulfed. Just like the amoeba engulfs its lunch, we engulfed, our ancestral cell engulfed some other cell. However, it found that that cell was being pretty beneficial to its life. The mitochondria, for example, was mass-producing energy, which is kind of a good thing if you're a cell looking to spend ATP in a variety of different things. So we talked a little bit about connections between plant cells. What I want to focus on now is connections between animal cells. In animal cells, we don't have a cell wall that's going to join up one next to the other. But we do still need a means of communicating information between the 2 different cells. We have specific pores available for that use in plants. We also have that in animal cells. What I've drawn up here are 2 epithelial cells. As you might imagine, they're creating a nice strong boundary between what is outside of this organism and what's on the inside. We've got other cells underneath this, right? These cells down below need to be protected by our epithelial cells. And so what we're going to have happen is that these epithelial cells need to talk to one another. They need to share resources. If one has a lot of amino acids, it needs to be able to pass it to the others. If one has a lot of salt, it needs to pass it to the others. And so we're going to have these little channels, and those channels we call gap junctions, because they're literally a gap in the membrane where these 2 things join. These gap junctions are going to allow substances to cross back and forth depending on where they're needed. And that's going to allow us to use diffusion to share resources rather than our energy requiring exocytosis. This is just the random movement of the particles, help them move around. We also want to make sure that other things do not move around, right? The stuff inside the cells that they need to share, we want to be able to share them. But the stuff outside the cell, up here, maybe it's bad for cells normally. And our epithelial cells are well adapted to deal with it. For example, in your stomach, you have hydrochloric acid. That acid would normally be quite harmful to cells, but the stomach cells, the ones lining your stomach, are prepared for it. If they weren't well connected, if they weren't stuck together really nice and tight, that acid might be able to drift down between the cells and reach these ones that aren't well protected. And so what we're going to do is create a very strict boundary line between these 2 cells. We want them to be held together really, really tightly to prevent that kind of leakage from getting to the tissue below. And so the types of sort of tools we use to hold these cells together are called either tight junctions or desmosomes. As with the gap junction, junction basically just means that we've got two things coming together. And so here we've got two cells coming together. A tight junction is going to tightly hold them together. And you can see it's sort of like there's a ton of little dots if you look at the diagram in your book. And basically what they're trying to demonstrate there is that these tight junctions hold these cells together really tightly to prevent leakage from here down to the cells below. We're also going to want to try and anchor these two cells to one another a little bit more than just at that sort of membrane area. So we build a second line of defense called the desmosome. The desmosome, you can see here, has projections that stick to either side. Those projections are made of intermediate filaments. And as you know, intermediate filaments are quite strong. They're composed of things like keratin. If you recall, keratin has that triple helix. And you take a bunch of those, string them together, you're going to get these strong rope-like structures that stick out into the cytoplasm of this cell and stick out into the cytoplasm of this cell. And so they're going to really hold this together sort of like a staple or like a rivet. So we've got tight junctions, we've got desmosomes, and we've got gap junctions. Those are the connections between cells like the epithelial cells we were discussing.